Hi, I'm Hirsch, and I'm a final year engineering and biomedical science student at Monash University. This year I conducted two projects in partnership with the Department of Physiology with Dr. Yan Wang and Dr. Nicholas Price. For the second part of my final year project, I was interested in investigating one of the fundamental questions in systems neuroscience. How can we characterize the codes with which neurons communicate information to each other? For our project, we analyzed a dataset from area MT in the visual cortex in marmoset monkeys. MT neurons represent the direction of a moving stimulus through their firing rate, in other words, the number of spikes that fire inside an integrating time window. What we investigated was whether there's additional information in the precise spike timing patterns inside that window. In particular, some neurons tend to produce short bursts of spikes closely spaced together, and we explored the relationship between this burst firing and direction of motion of the stimulus. We formulated two project aims. Firstly, can we identify the phenomenon of bursting in MT neurons? To identify bursting neurons entails finding those that output spike bursts significantly more often than chance. To do this on a per-neuron basis, we implemented a trial shuffling algorithm which scrambled the within-trial spike correlations. We could classify which neurons had bursting activity based on whether they burst more often than their shuffled responses. In our dataset, we classified 48% of the MT neurons as bursting neurons. Our second aim was to determine whether these bursts provide information about the stimulus direction better than all spikes together. We trained a set of linear classifiers to decode the stimulus direction from the MT neuron data. What we found was that the burst rate is significantly less direction informative than the spike rate over any time window. In general, this tells us that bursts have a lower signal-to-noise ratio for direction coding in area MT. And given the sparsity of research into burst coding in the visual cortex, this project is novel in its information-theoretic approach to visual cortex bursts, and our finding is also novel. In this video, I'll discuss in depth our methods, the conclusions we can make from this finding, and how this approach could be taken further. To begin, it's worthwhile to run through the basic mechanisms of neurons, action potentials, and stimulus coding. Neurons are specialized cells of the brain, which communicate information to each other with patterns of electrical activity. Fundamentally, the language of this communication is based on the action potential. This is essentially a brief spike of membrane potential about 1 millisecond in duration. Action potentials, or spikes, propagate down the body of a neuron and are passed on to other connected neurons. If we record the extracellular voltage of a neuron, we might see something like an infrequent series of spikes, where a new spike occurs on average, let's say something like 10 to 20 times a second. This kind of behavior is called a Poisson process. The exact intervals from one spike to the next aren't fixed, and in fact timing of each spike is independent from the previous one. But at any given time, the underlying average rate of spike firing is steady and doesn't depend on when the last spike occurred. This firing rate is the primary metric through which neurons convey information. For example, one well-characterized area of the visual cortex is the middle temporal area, also known as area MT. Neurons here are sensitive to the motion of a moving stimulus. If a line crosses an MT neuron's receptive field at its preferred direction, its firing rate increases. At its non-preferred direction, its firing rate drops. The exact timing of spikes in response to a repeated stimulus is highly variable, but the rate is more steady. We describe this behavior as rate coding, and while it's certainly real and demonstrable, there's reason to believe that the rate code isn't the whole story. For instance, we know that many neurons don't exactly follow the model of the Poisson process that I described earlier. To illustrate, compare the two neurons here. Both have the same average firing rate. And in the first, the spikes are spread out in the distribution we'd expect from a Poisson-like distribution. In the other, we observe that the spikes are more clustered into small groups that are separated by longer intervals. We call these bursts, and they occur commonly in neural recordings. In this case, the spikes in each trial aren't independent, they're correlated. Clearly, we're more likely to see spikes around other ones than by themselves. This observation of bursts is closely tied to the study of another type of neural code called temporal coding. For the rate code, we're concerned with measuring a neuron's average spike rate, which implies that we integrate the total number of spikes inside some time window. The concept of temporal coding implies that the timing patterns of spikes inside this window, such as the presence of bursts, is also relevant for information encoding. There's a few ongoing theories as to the importance of burst activity in neural coding. One is the theory of multiplexing, that bursts represent another channel of information where they encode different stimulus features than single spikes. Another hypothesis is that bursts represent the same information, but they're better at it. The idea here is that single spikes are a noisy information channel, so bursts may be the neuron's way of sending stimulus features with a better signal-to-noise ratio. If this is true, then we'd expect that if we take a population of bursting neurons, we get the best information about the stimulus by looking at the bursts alone. This provided an opportunity for my project to firstly investigate the presence of bursts in area MT neural data, and then their contributions to direction coding. We used a dataset of 197 MT neurons, spliced together from 4 anesthetized marmosets, recorded during the presentation of random dot motion stimuli over 12 directions. For the first step of measuring the bursts in neuron activity, we need a formal definition of what constitutes a burst. The simplest definition is a continuous group of spikes where we define a parameter for the maximum time interval between consecutive spikes. In other words, for each spike, we measure the preceding inter-spike interval, and if it's less than the chosen threshold, then it constitutes part of a burst. Previous burst studies in the visual cortex and elsewhere have somewhat varying selections for this threshold parameter, but a value that's well precedented is 8 milliseconds. Our next step is to determine, for each of the 197 neurons, whether they could be classified as bursting neurons or not. Essentially, all neurons will produce some quantity of spikes that fall within our 8 millisecond threshold, so our task is to verify which ones do this more than standard Poisson spiking neuron would. Going back to our spike plots from earlier, in the first Poisson-like spike process, if we were to measure all of the interspike intervals, or ISIs, and plot these intervals onto a histogram, we'd see a distribution like an exponential curve. This is the nature of any Poisson distribution. In the second process, the average ISI is the same, but the presence of bursts changes the shape of the ISI histogram. There's a larger peak here that corresponds to burst spikes, with a plateau that conveys the isolated spikes in between. To verify which neurons exhibit ISI histograms that look more like the bottom figure, we implemented a trial shuffling method that works as follows. We take a neuron set of responses for its preferred direction and divide the 500 millisecond stimulus motion period into 10 millisecond windows. Inside each window, we shuffle the spike times across trials so that each trial is left with the same number of spikes as it did before. The timings of those spikes are now randomly pulled from other spikes in that window. What this process does, essentially, is discard the timing relationships between adjacent spikes, while preserving the average firing rate and the average ISI for that neuron's responses. For this sample bursting neuron here, if we plot the ISI histogram of the original trials and the shuffle trial set, we see that resampling dissipates the burst peak. Our burst proportion, which is the ratio of spikes to the left of our 8 millisecond threshold, has decreased. We run this shuffling algorithm a large number of times, at least 1,000, and verify that the original burst proportion was greater than in the shuffle trials, at least 95% of the time. Using this criteria, 94 out of our 197 neurons, or about 48%, are classified as bursting neurons. Our next step is to determine the comparative direction coding in bursts compared to spikes. To do this, we see how much of the stimulus direction we can decode from each. We train a set of linear decoders to calculate the stimulus direction using linear discriminant analysis. This exploits whatever correlations are present between spikes in direction or bursts in direction, and it's a simple and direct way to see how much information about one is contained in the other. The first decoder is trained on the spike rates of the neurons. The second decoder is trained on their event rates, where an event is either a burst or an isolated spike. The third decoder is trained on the neuron's burst rates. We ran each decoder on the 94 bursting neurons, as well as on randomly sampled populations of 94 neurons. Decoder performance was measured over increasing time windows of data, from 100 milliseconds after motion onset. The primary observation is that burst rates are significantly less informative than the spike rate in bursting neurons or otherwise. This is contradictory to the hypothesis of bursts having higher signal-to-noise ratios, and it tells us that single spikes increase the level of information in the neuron. We can deduce that bursts overall are a poor vector for direction information but what our analysis doesn't examine is how much information is in a single burst compared to each single spike. Given more time, this would be a worthwhile quantity to investigate, although a different approach would be required. Instead of stimulus decoding, this could be accomplished by measuring the amount of mutual information between a spike or burst and the stimulus direction. Also, in this project, I've focused on direction coding because that's a primary stimulus attribute that area empty neurons are selective to. However, there's scope for further research based on other stimulus properties in area empty or in other areas of visual cortex. In particular, it'd be interesting to investigate burst coding for time-dependent stimuli with changing contrast and direction, and compare how bursts respond to the dynamic content of stimuli compared to the spike rate. One of the central challenges I faced with my analysis was in ensuring the validity of my results in terms of determining when results actually described meaningful differences between spikes and bursts, or when they were just confounds of some factor that I hadn't controlled for. For instance, at one point I plotted direction tuning curves for each neuron. These are graphs that show the strength of neuron response against all stimulus directions. I found that bursts and spikes produce different curves in terms of width or circular variance, but it was unclear whether this represented actual information differences. To determine whether the difference between curves was meaningful, I ended up running the same analysis on the trial shuffle data, where any bursts present are just coincidental, and the same result appeared. This implied the difference in tuning curves wasn't an inherent property of neuron bursts, but just statistically expected. So I've taken this as a lesson on the importance of applying the right statistical controls. A significant part of this field is based on analysis of microelectrode cortex recordings, and with my project A and B, I've learned a great deal about these methods of analysis, how we can take electrical recordings and turn them into neuronal characteristics and relationships. Altogether, this project has built on my earlier experiences in this lab and helped me to develop a foundational set of knowledge and skills in neurophysiology.